Let me start with a simple question. What if the most important diagnostic test in medicine wasn't a test at all? What if instead, it was a foundation, a single, comprehensive readout of the human genome that everything else builds on? Because that's exactly the shift we are witnessing right now, and the inflection point became very clear at the AGBT 2026. For the past decade, precision medicine has been incredibly successful, but also fundamentally constrained. We built our entire system around targeted testing, small gene panels, single biomarkers, one drug, one diagnostic. And it worked, but only within limits. Because every time we asked a biological question, we had to decide in advance what we were looking for. That meant missed variants outside panels, tissue exhaustion, sequential testing delays, and ultimately, incomplete answers. So we optimized within a box, and for years, the industry accepted that box. Then came a signal strong enough to break it. At AGBT 2026, Roche introduced Axelios One, a new sequencing platform built on SBX technology. But this wasn't just another sequencer launch. It was a statement that a whole genome could cost around $150. It could be generated in hours, not days. It could deliver clinical-grade accuracy and even capture multi-omic signals like methylation. Now, let's be clear, this wasn't happening in isolation. Illumina had already built the dominant ecosystem. Ultima Genomics was pushing cost disruption. Pacific Biosciences and Oxford Nanopore Technologies were redefining read lengths. But for the first time, one platform started to converge cost, speed, accuracy, and clinical intent. And that changed the conversation, because when cost collapses and speed accelerates, something fundamental happens. The default changes. We are moving from panel first gives to WGS-first, reactive testing gives to proactive profiling, single snapshot gives to longitudinal monitoring. In this new world, you don't ask, which test should I run? You ask, how do I interpret the genome I already have? And that is a completely different paradigm. Nowhere is this more powerful than in companion diagnostics. Imagine a world where one assay detects SNVs, indels, copy number changes, structural variants, tumor mutational burden, MSI status, all at once, and not just once but continuously, across the patient journey. This unlocks WGS-first CDX in oncology, faster rare disease diagnosis, unified assays for clinical trials, real-world genomic monitoring. The CDX is no longer a test tied to a drug. It becomes a platform tied to the patient. But here's the reality. Technology is moving faster than the system around it. We face real constraints. Regulators still expect to find locked assays. Clinicians need clear, actionable reports. Bioinformatics pipelines must scale massively. And reimbursement still lags behind innovation. Most importantly, sequencing is becoming easy. Interpretation is not. The bottleneck has shifted from generating data to understanding it. And pharma is already adapting. They're not asking for more assays. They're asking for one global, standardized platform, faster turnaround, ideally under 48 hours, multi-biomarker readiness, and integration with real-world data. Because for them, the goal is simple: reduce complexity while increasing insight. So what does this mean for diagnostic companies? It means we have to fundamentally rethink who we are. We are no longer just assay developers or technology adopters. We must become platform orchestrators, data integrators, interpretation leaders, because in the near future, sequencing will be commoditized. But the ability to turn genomic data into clinical decisions that will define the winners. So let me end where I started. What if the most important diagnostic test wasn't a test? What if it was the genome itself, captured once, interpreted continuously, and applied across a lifetime? That future is no longer theoretical. It's being built right now. And the real question is not whether it will happen. The question is, who will lead it?